Yeah, students. So we just recap the last session contents. So I just ask you a few questions regarding whatever we discussed at present. So virtual screening using Autodoc VARX. Good. Okay. The first question is, what are the extension of ligand and receptor files we consider for docking in Autodoc? Okay. So what are the extension of ligand and receptor files we use in docking through Autodoc? We have so many extensions of files, isn't it? Generally, we have to use 1D file, 2D file, or 3D file. If it is a 3D file, what kind of file extensions Autodoc support for docking? Cybel Mol 2. So this is actually a raw input file. Cybel Mol 2 is a raw input file, but ultimate file. If you have that file, there is no need of any conversions. We can reduce a few steps, intermediate steps. That's the ultimate extension of files we use for docking. What is that? Yeah, only one has responded. How about the others? Right or wrong, you just respond. You can check, okay, whether you know or not. So there is nothing wrong. No one is going to grade, even at the last time, even at the last session. You just that is for self-assessment. Okay, so no grades are going to reflect in the certificate. So why can't you try? What are the extension of files? So only one participant is here for interaction, is it? Good, very good. Another has tried, good, PDB. But this is not the exact answer still. Not, not. Very good, tried well. Superb, STF, good, tried well. But this is not the correct answer. Very good, try. Other participants also can try, even anyone can try with any answer. But at least you have interacted. This made me to proceed further. We use PDBQT files. Okay, this is the ultimate file we create either from Autodoc Mina, Autodoc 4.0, Autodoc PYRX. Okay, so these are the files we convert, and then finally we go for running the grade and running the doc in Autodoc. This is how we make the steps. It can be of any Autodoc versions. Okay, now let me go to the next question. So that is related to the force fields. Can someone provide me the expansion of charm? There is one force field called charm, isn't it? I want the expansion of charm. So you can either unmute an answer or you can type the answer. This is the most important force field used by most of the computational biologists for MD simulation. So this is energy minimization. This is actually used for energy minimization protocol. Okay, tell me the expansion. Anyone remember? You try. Meanwhile, I can show you. So it is actually good, superb, excellent. Chemistry at Harvard, molecular mechanics. You're right. So likewise, I want four examples for uh, force fields. Four examples. Any four. I already provided charm. Other than charm, you can provide any other four. Good. OPLS, amoeba. Good. Amber. CFF. No PFF. Polarized force field. Consistent force field is there. Okay, good. You have tried well, but if you go for classification of force fields, you remember how we classified on yesterday. That CVFF is also there. You remember how we classified the force fields? We have classified like classical, polarizable, reactive. So under this, we have seen some examples, isn't it? Good. Also remember this. Okay. Now let me go to the no Chromax is a software. You can't tell it as a force field. In you can use Chromos. Chromos we can consider. Okay, Chromax is a software. Chromos is a force field, is for energy minimization. Okay, now let me go to the next question that is related to MD simulation. Give me any 4 to 5 analysis of MD simulation. Any 4 MD simulation analysis. If you want to analyze the simulation output, okay, how we can analyze the MD simulation? Very good. The first answer is RMSD. I think everyone remember the expansion of it. Root, mean, square, deviation. So likewise, I want RMSD is the correct answer. Likewise, go with other answers. Good. RMSF, root, mean, square. Root mean. Um, yeah, of course, square fluctuations. Okay. RMSF, root mean square fluctuations, radius of variation, RG. Good. Any other? You remember the energy, surface area, hydrogen bond interaction, secondary structure predictions, etc. Do you want me to show? Yes. If you see here, okay, I also shown you a kind of a binding free energy determination, MMPBSA, MMGBSA, LIE. This is related to one trajectory energy or three trajectory energy, or you consider two trajectory energy. For these different formulas are there, isn't it? So other than this, we have seen this is a kind of a secondary structure prediction. Okay. And this is actually a surface area versus time. Okay. This is another kind of a plot. Okay. This is likewise, what else we have seen? Yeah, I think I have shown very few, but also we have uh, analysis, principal component analysis. If you have more fluctuation in RMSF, okay, you better go, what I told, actually root means square deviation. Here I told like root mean square deviation and root mean square fluctuation is right. So in the fluctuation, okay, if you have more fluctuation, then you have to consider working on principal component analysis only for these residue positions. Okay, the docking of your ligand with the receptor is having some kind of deviation. So this you can check with the PCA. Okay, there are plenty of analysis. Okay, 10 to 15 analysis are there, but most important analysis alone I have explained. Very good. So I found interaction from most of the participants. So let me then go to the chapter 8. The chapter is about the basics of cancer biology, genomics, and epigenetics. The our theme is to work on AI and machine learning approaches of and vaccine design in cancer, isn't it? So we have to know about how to design the drug, now how to design the vaccine in cancer. So one should know first of all about the basics of these three concepts. Let me take you to the cancer. What is a cancer? When you can tell if cancer has occurred in a particular organ. Actually, it is a word derived from a crab, that is cancer, derived from a Greek word for crab, that is carcinoma. In Greek, for the crab, we call it as a carcinoma. So the cancer came into existence. That word coined from that. And if you see, the cancer is a group of diseases involving abnormal cell growth with a potential to invade or spread to other parts. So the cancer, if you find the liver cancer, okay, that doesn't mean that it's going to affect only the liver. Even after some time, it can go for metastasis. Even the abnormal cell tissues proliferation from one organ can migrate to the another organ, and it can also try to make these kind of abnormal cell proliferation in that specific organ. So you can find even the metastasis happen, okay, through the traveling of abnormal cells through the blood stream. And if you see this kind of this contrast with benign, which do not spread, okay. So in the benign tumors, we don't find any metastasis. Okay, only in malignant type you can find the metastasis. So this is of course one of the difference between benign and malignant tumor. If you see the types of cancer, okay, the cancers can be of carcinomas, sarcomas, leukemias, lymphomas. Okay, carcinomas. I am basically working on carcinomas. Actually, a carcinoma begins in the skin or the tissue that covers the surface of internal organs and glands. Okay, and so it usually form the solid tumors, and these are the most common type of cancer. Example: prostate, breast cancer, lung cancer. So if you see lung cancer, we call it as a lung carcinoma. If you see in liver, we call it as a liver carcinoma. Okay, so this is actually occurs in the outer surface or just in the tissue, not in deep. And if you see the sarcomas begin in the tissues that support and connect the body. That is, it is generally developed in fat, muscles, nerves, tendons, joints, blood vessels, lymph vessels, cartilage. So, if a patient is suffering from sarcomas, it's very, very difficult to treat when compared to carcinoma. And leukemia, this is the third type. It is a, of course, cancer is occurring in the bloodstream. You know, there are more difficulties if a person is suffering from blood cancer. But still, the 5-year survival rate is increased in blood cancer. So how this can be done? So 6 liters of blood in here in a 75 kg male or female, okay, is going to transport from the head to the toe. So if so, if the entire blood is infected with cancer, how this can be done? They do the kind of they go for the blood kind of transplantation. Okay, so they not take or the entire 6 liters of blood, but they take 1 liter of blood. Okay, they remove the 1 liter of cancer blood tissues and then they inject 1 liter of healthy blood. And the next day, they again remove the 1 liter of blood. Okay, I think even during the blood donation, we can donate up to 330 ml, isn't it? So even if you take that 330 ml, so the 330 ml alone is done through various uh, time periods, and they try to remove as soon as possible. So it take even 1 year to 2 years even for the treatment itself, based on the patient condition. But this is of course a curable. Okay, and if you see the blood tissue, you have numerous cells. Okay, you have WBC, RBC. In WBC, you have lymphocytes, leukocytes. You have granulocytes. So much cells are there. So all these cells has to be changed. Okay, from the cancer patient to the this has to be changed from the healthy patients. And if you see this here, you can have types like acute lymphotic leukemia, chronic lymphocytic leukemia, acute myeloid leukemia, and chronic myeloid leukemia. So, and if you see the fourth category is the lymphomas, basically it occurs in the lymphatic system. That is on the network of vessels in the glands that help fight infection. Okay, so there are actually two types of lymphomas. One is Hodgkin lymphoma, the other is non-Hodgkin lymphoma. So if you see the basic differences between the benign and malignant, so benign, it grows slowly, but in this cancer type, it grows rapidly. Here it is, we find a well-defined capsule, but here they won't find not capsule. And this is an invasive, the malignant is invasive, benign is not invasive. So you don't find any metastasis in nearby organs. And if you see benign is well differentiated, but malignant is poorly differentiated. Low mitotic index, high mitotic index. Do not metastasize, can spread distantly metastasis. How does cancer form? We already discussed. It is a disease caused when cells divide uncontrollably and spread into surrounding tissues. What causes cancer? The cancer is caused by changes in DNA, isn't it? Most cancer causing DNA changes occur in sections of DNA called genes. So this is actually a genetic change of this is occurred because of the genetic change. And if you see what are oncogenes, a DNA change can cause genes involved in normal cell growth to become oncogenes. Unlike normal genes, oncogenes cannot be turned off, so they cause uncontrolled growth. What are tumor suppressor genes then? So actually, in normal cells, tumor suppressor genes prevent cancer by slowing or stopping cell growth. The DNA changes that inactivate tumor suppressor genes can lead to uncontrolled cell growth and cancer. So one should know about all these technical terms before going to working on various genes on cancer. What is a tumor microenvironment? So here you can see the diagram. Here in normal cells, tumor suppressor genes prevent cancer by slowing or stopping cell growth. The best example you can take tumor necrosis factor. That's the most important protein, okay, tumor necrosis factor that comes under a classification of a cytokine, which is going to combat the cancer. Okay, even in tumor necrosis factor, you have subclasses to FTNF alpha, beta, gamma. Generally, you see TNF alpha is majorly combating all the types of cancer cells. And if you see this DNA changes that inactivate tumors, if you find TNF cytokine is low, obviously the person is more prone to get this kind of prion diseases, especially cancer diseases. How does the immune system interact with the cancer? If you see immune cells can detect and attack cancer cells. Not only the proteins like tumor necrosis factor can combat the cancer cells, but also you find the immune cells like the T cells, especially cytotoxic T cells. Okay, so these directly go and combat. And T helper cells take help, okay, of course, to combat the cancer. Likewise, you have cytotoxic helper, and you have natural killer cell, okay, and of course, even the antigen presenting cells like B cells, uh, dendritic cells, macrophages, all these cells try to combat the cancer. And if you see the B cells can also produce humoral immunity by producing more antibodies. Okay. So if you get a kind of an antigen recognized by the antibodies, even the antibodies produced by the B cells try to combat the cancer. How do genetic changes affect cancer treatment? Each person's cancer has a unique combination of genetic changes. Specific genetic changes may make a person's cancer more or likely to respond to cancer treatment. Okay, a DNA change. So only a small change can lead to a cancer. And of course, this can also be rectified, okay, during the genetic counseling. So before going for the marriage and coming for the birth, okay, uh, then even the most of the developed countries like US, okay, they go for genetic counseling if they have some hereditary kind of a illness with respect to cancer. They try to identify the genes and try to rectify, okay, before going for giving a birth. What causes genetic changes? Heredity, UV radiation. This is most important, of course. Even we go for creating we irradiate the tissues, okay, of course, to make normal healthy cells to the cancer cells, and we do even research on this cancer cells. So, radiation is, of course, used to convert the cancer cells in the form of cancer cell lines. Chemicals. Okay, so many carcinogens. Okay, especially even the chemical, nearly hundreds of chemicals are present in cigarette smoke. This can cause cancer, especially NDEA, N-nitrosyl diethylamine, 4-amino biphenyl. These are present in excess in cigarettes. So this can cause us cancer. And viruses, I think we have also seen. in the earlier classes, okay, where there is a strong relation. You have a strong relation between virus and the cancer. That we call it as a viral etiology, isn't it? So if you see the hepatocellular carcinoma is majorly caused by hepatitis viruses, and if you see cervical cancer, you can find more human papillomaviral infections. Likewise, a few cancers are more strongly associated with virus. So if you have 99% of, if you have a infection, 99% infection, more even uh, you can get 100% of cancer. Okay, so it means that most directly proportional if the patients are infected with the viruses. Cells dividing capacity. How much cells you have, what is the hemoglobin count, and how about the other cells? Okay, so all these things. Okay, what is the mitotic induction? That based on that also you can find the spreading of cancer. How does age lead to cancer? Of course, during the aging, we can find a small change will be there in the DNA, and so this can also lead to a higher risk of cancer. The 5 year survival rate will be in, 5 year survival rate will be decreased if you go for the senior persons. But even in adult, you can find they can combat many types of cancer. But even the child is you can find the aging can lead to a reduction of 5 year survival rate. So 5 year survival rate is less in child and senior when compared to adult. What is metastasis? So just now I described, okay, like cancer cells can break away from the original tumor that is from a particular organ and travel through the blood or lymph system to distant locations in the body. Okay, so I came to know the person who is infected from spinal kind of a cancer. Okay, he can't able to find. He didn't find any kind of a symptoms. Not only the spinal cancer, even the other types of cancer, it's very difficult to recognize with the symptoms. Actually, the spinal cancer, it just spread, okay, from the back to the front. It infected the heart, and it infected the lung and liver. So they don't understand, even doctors, what is the exact reason. Then they finally diagnose, okay, so you are getting a chest pain, okay, just because of the spinal cancer, which has already spread in the three to different, 3 to 4 different organs. So it's very difficult, isn't it? If you feel pain, okay, for example, if a person is infected from colon cancer, definitely you feel pain in the stomach, okay, stomach region. But it's easy if the cancer is occurring in the digestive tract, but it is very difficult if it is somewhere like in the spine or in somewhere in the pancreas. Types of cancer studies done in my lab. Actually, I worked on ovarian cancer. Okay, so how to treat the ovarian cancer? It published in Elsevier journal. So that is these kind of researches happened in the last 3 years. Kidney cancer, I just diagnosed like if a person is suffering from second stage of kidney cancer, that can be cured. But if a patient is moving to the third to fourth stage, that itself is very difficult. Obviously, they have to go for kidney transplantation. So in that case, okay, so if I can able to cure, if we can able to cure for the first or second stage, what are the important genes responsible, okay, for the stage 1 and stage 2 kidney cancer? So I've identified through various analysis. Almost I unraveled all the genes present in kidney cancer. This published in scientific reports. In the subsequent slides, you can find how I worked on the diagnosis of the kidney cancer, especially stage 1 and stage 2. And hepatocellular carcinoma, how to prognosis, diagnosis, and treatment of hepatocellular carcinoma. Also, this I worked extensively, and also how to treat the pancreatic cancer. Now, if you see therapeutic targets for the hallmarks of cancer, there are numerous inhibitors, molecule inhibitors used for preventing the cancer. Okay, for example, if you see the EGFR inhibitors act on sustaining proliferative signaling, and cyclin-dependent kinase inhibitors evade growth suppressors. So each and every inhibitor has a mechanism of action on the treatment of cancer. So this we call it as a hallmarks of cancer. Okay, so this is a popular picture you can see. Anyone is going to describe on how to treat the cancer. So it's very easier for us. Okay, if you go for drug design with the small molecules. Okay, if you have a plenty of protein targets, these are protein targets, and of course, each and every protein target can lead to a pathway, an individual pathway. Okay, if you want to target a VEGF pathway, then obviously you have to design an inhibitors, which in turn can induce the angiogenesis. So likewise, you have plenty of inhibitors and mechanism of action. Now come to the cancer chemotherapy. If a patient is infected from cancer, diagnosed with the earlier stages of cancer, obviously he or she will be subjected to chemotherapy. Okay, chemotherapy is of course painful, but in the initial stages, it can give a good remedy. Okay, in combination of cancer chemotherapy, the doctors can also suggest to take the radiotherapy. So these two are generally done for the cancer patient, for any type of cancer patients. If you see the cancer chemotherapy, here also you can find the major classifications like even the anti-metabolites like purine and pyrimidine inhibitors, okay, can be used, okay, for treatment of cancer. So generally for the anti-metabolites here, we can take examples like antifolates and miscellaneous. Likewise, we can see a different kind of chemotherapy. Anti-metabolites is number one, and other molecules like vitamins and alkylating agents, topoisomerase inhibitors. So these are some of the things, and even the mechanism of action is described here. Now we can see each and every inhibitors we use. Okay, so first is mitotic inhibitors. The examples are taxanes and alkaloids. Okay, actually, of course, this information is present in cancer biology textbooks. We have plenty of textbooks. Okay, from the basic textbook of cancer biology, just take on the extract. Okay, so you can find not only the examples for a particular classes of drugs, but also mechanism of action. Here, if you see the mitotic inhibitors, like alkaloids, taxanes. Okay, so alkaloids actually work on microtubule polymerization. But whereas the taxanes, okay, this is another class of mitotic inhibitors. It act on microtubule degradation. So one is going for microtubule polymerization, the other is going for microtubule degradation. Okay, this mitosis can block and provide cell death to the cancer cells. Okay, so this is an example for mitotic inhibitors, and you can find more many therapeutic applications. Like it is going to treat prostate cancer, breast cancer, neck cancer, head and neck cancer, Kaposi's, sarcomas, lung cancer, etc. Now let me go to the second classification: alkylating agents. In alkylating agents, we can find platinum compounds, nitrosoureas, nitrogen mustards, triagenes. Okay, these are the classes, major classes of compounds which or inhibitors. We use it as an alkylating agents for these therapeutic applications. You see, the alkylating agents, okay, is going to have an alkyl group in the compound. This alkyl group, okay, can go for DNA. The alkali, this is going to uh, go for DNA alkylation. This alkyl group go for alkylation process. Obviously, it combat only the cancer cells. So the multiple DNA strands, which helps us to proliferate the cancer cells. Okay, this is going to die obviously, and obviously the cancer cells can go for apoptosis and cell death. Okay, so this is something related to alkylating agent. So it facilitates the DNA alkylation to inhibit the cancer cells to kill, so go for apoptosis in cancer. Now, if you see the third type, anti-metabolites. Okay, in anti-metabolites, we find pyrimidine antagonist, purine antagonist, antifolates, enzyme inhibitor. See, from the beginning, I was explaining you about the classes of inhibitors, the types of inhibitors used for inhibiting the cancer. We have plenty of inhibitors, isn't it? I'm just completed the three, and now I'm going for the fourth called anti-metabolites. So if you have already existing drugs, okay, and you also have a different mechanism of action, okay, then why we have to go for cancer drug discovery? You know, 55% of the drug discovery in the world is happening in treatment of cancer disease. Even myself, I'm presently working on two funded projects, or both of funded projects are in cancer, anti-cancer drug discovery. Not only that, the past 3 years I got funds, okay, not only two funded projects, three funded projects on cancer. And if you see even the training programs, I have received funding from the DBT, CSAR, ACTE, so many Government of India funding organizations to conduct training on cancer. Okay, in the last 3 years, I received three funded projects and three training programs. I mean, from the government funding agencies. So we have a lot of. So what you can understand, so if you want to treat a cancer, okay, I mean, if you want to treat the cancer patients, obviously one should have a knowledge on personalized medicine, precision medicine. You can't directly give like a fluorouracil and you can tell this can able to cure breast cancer completely. You can't tell, you can't guarantee it. Even this drug can able to cure one out of four patients or one out of three patients, not more than that. If so, then how this can be treated? Obviously, you have to go for tailored medicine. So one day, this certificate course is designed in such a way that even to go with the existing drugs, one should have a knowledge on even how to go with the existing drugs can be used for repurposing. Okay, and also how to modify the drugs. You remember, I explained you about fragment-based drug screening. Even if you modify some functional groups, you may increase the efficacy of a drug on treatment of a disease. So all these things play a very important role. Of course, small molecule inhibitors are the major drug molecules dominating in the industry, even in combating cancer diseases. Even for the cancer diseases, many people go for taking the small molecule inhibitors. But the effective remedy is to go for specific antibodies. The antibody is going to have specificity, and of course, it can also give an important role in personalized medicine. Okay, okay. Let me go to the anti-metabolites. So, if you see the anti-metabolites, okay, so the mechanism of action, purine, pyrimidine antagonists can work on DNA incorporation, H, hypoxanthine guanine phosphoribosyltransferase transferase. You remember this is an enzyme important for construction of MABs. If you want to develop an antibodies, okay, monoclonal antibodies, you have to rely on this enzyme HGPRT and thymidylate synthase. Okay, so this antagonist can work on this kind of a mechanism. Okay, one is for the DNA incorporation, the other is for producing the MABs. Okay, sorry. And likewise, we have a different mechanism of action for the antifolates and the other enzyme inhibitors. So DNA crosslinks are formed, DNA telomerase is happening on. So obviously, the cells are not going to proliferate. The abnormal cells is not going to proliferate. Now, obviously, can fight because of the mitosis not going to happen. Obviously, the cells are going to die and anti-tumor antibiotics. These are, of course, another classification. Here you can see the mechanism. I told you already about that these detailed information are available in plenty of cancer biology textbooks. So the other is topoisomerase inhibitors. Now, something about precision medicine design in cancer. So actually, if you want to go for precision medicine design, okay, one should know about the different categories, okay, of bringing this precision medicine. Actually, if you see the types of trials, the number one, we can see the basket trial. Here, you can see three patients are there. Okay, might be a patient is suffering from breast cancer, the green is suffering from a liver cancer, and the blue one is suffering from lung cancer. Think like an assumption, if three patients are suffering from three different cancers, that's what they mentioned, like multiple diseases. But all these three patients may affected by a single gene. can be EGFR, epidermal growth factor receptor, or can be heat shock protein. It depends. Okay, so we can't tell that these patients are infected from cancer because of a different gene. Even it can be a single gene. Then how these diseases can be, how we can target only the specific organ. Okay, that's most important, isn't it? So one should have a knowledge on that. Okay, on targeting the interventions in the basket trial. Now go to the umbrella trial. This is the second case of treatment. Okay, so in precision medicine, you should come across the different categories of treatment. The second is umbrella trial. Here, if you see, there are three patients, but all the three patients are suffering from a same disease. For example, all the three patients are infected from breast cancer. Okay, if they have breast cancer infections, then obviously you can't tell all the three patients have are infected from a same gene. One might be a BRCA1, the other might be a BRCA2, the third can also be from the PEGFR, vascular endothelial growth factor receptor. If these three patients have diagnosed with three different genes, then the drug, whatever they're going to, the doctor has to initially diagnose the gene, they have to identify the gene for causing, and then he has to treat only accordingly. He has to provide the inhibitors only according to the gene. Okay, can't give a same drug like if anyone is suffering from fever. Okay, anyone will try to prescribe paracetamol. Isn't it? The clinicians try to prescribe paracetamol, but that is not the case in cancer. In any cancers, first we have to identify the gene, then only we have to even after identifying the gene, even you have to check even what are the other genes, uh, for 5 to 10 genes, top 5 to 10 most interacting genes to the main gene. Then even we should see whatever drug we take, it target even the interacting 5 genes or 10 genes, whatever you're taking in study. Okay, so this is actually extended to system biology approach, like identifying the gene interaction networks, or if you go for protein interaction networks, all these things. Okay, so this umbrella trial can also lead to the system, can extend a field called system biology to get a precision medicine for cancer. Platform trial, this is the third type. Here, if you see, okay, you find the types of analysis, interim analysis. See, if you have a patient, okay, here you have a standard care for a patient. Here, one is intervening, and there are also subclasses of different trials. Here also, it can go for cohort study, retro study. There are types of studies. These are actually find in research design. So you have categories of research designed for bringing a drug for the treatment of cancer. So one should have some knowledge at least about the basic information on basket trial, umbrella trial, and platform trial. Here, platform, here it mean like generally, if you see the cancer hospital, many patients come for treatment for many diseases, many cancer diseases, isn't it? So then, if they come, okay, so well, what kind of a platforms like if a patient is suffering from respiratory illness, obviously he has to taken to the pulmonology. If he is suffering from kind of the blood related thing, then he has to take to the hematology kind of a department, isn't it? We have different departments. And of course, the intervention, even if they is suffering from blood cancer, he has to also check for the other organs. So one has to check multiple diagnosis. I mean, with the different departments. Okay, so multiple departments comes into play. Okay, for the treatment of even a particular cancer. So we try to work on platform by platform. Okay, so for one cancer might be two departments important. For when two departments can play a role, but and another cancer we may find nearly 4 to 5 major departments can also play a role. So one should also able to find a better kind of intervention, okay, from the platform trials. Now we can see that actually there are so many case studies. Okay, I was explaining about cohort studies, like one person is suffering from lung, kidney, ovarian cancer, okay, brain tumor, rare cancers. Okay, there are types of cancers. So we have to check. Okay, so if a patient is suffering from lung cancer, might he also will be checked whether he is having any kind of metastasis or not, linked with, I mean, which is close to the lung. Okay, so all these things we can do through various styles. Actually, for precision medicine designed for a cancer, oh, wow, if a patient is infected from the cancer, and the better thing what the clinicians can suggest is to go for genomic profiling. Yeah, the patient has to undergo genomic profiling of a particular tumor tissue. Okay, if for a tumor tissue, if nearly 4 to 5 chromosomes or the major chromosomes, then that he has to go for the specific chromosomes also. There is no need to go for the entire genome. Even he can sequence up to the chromosomes, the most important chromosomes for the and then he can try to find what is the origin. Okay, and of course they have to go for biomarker analysis. So we have, if you see the hepatocellular carcinoma, we have ALT, AST. That is, these are the marker enzymes which will tell whether a patient is suffering from cancer or not, liver cancer or not. Even the stages of cancer can be identified somewhat through the biomarker analysis. So, like whether he is facing a benign or malignant. Okay, all these things also we can able to find, including acute type of cancer. And if you see the treatment strategy, so the patients are provided with a certain kind of inhibitors, okay, for treatment of cancer. So even they can give a combination of inhibitors also to treat a disease. So the clinicians diagnose initially, and then they can suggest you what kind of treatment they can undergo on monitoring and follow up. So of course, lot of interventions are most important for precisely treating a cancer. So these are some of the important factors like advancing cancer research, tailoring treatment strategies, prediction of treatment, targeted therapies, tumor classification, subtyping. You know, most of the cancers have 5 subclasses, 4 to 5 subclasses. One should understand about the stage of cancer up to, even the treatment is up to what stage of cancer. If that is, if it is crossing third or fourth, obviously, defaultly we understood they have to undergo transplantation. There is no other way. Okay, so the only the transplantation can be able to save the life. And implications for healthcare professionals. So yeah, now let me take you to the some kind of gene information. If you see, if you want to know about the cancer, okay, and the cancer gene expression for a particular cancer, I think even on the first day lecture, I showed something about oncology databases, isn't it? So mycancergenome.org. Simply type the disease name alone. You get a series of genes. You have to simply click that. You can to find the gene expression level. Like this DBMHCC. Just recapping you. OncoDB.hcc, and then webs.iiit.edu. So this can also give you biomarker analysis, and even you can download the data set, and you can also work on computation analysis using this data set. CBio portal can give you a plenty of info data related to a cancer. Here, if you see in a data set, you find 377 patients, okay, and 379 samples, okay, what taken, okay, might be two healthy volunteers, the other are the cancer tissue samples. They worked on it, and if you see here, you can find TP53. This is the most significant gene from the 379 samples. You know, 30.8 percentage of the HCCs occurred because of TP53 mutations. If you see the mutation, 118 mutations are observed. So more details on each and every gene and type of mutations and how much frequency occurred. All this information are available in C Bio portal. and you also find the survival rates, mutations, and how the counts. More information you can find in CBio portal. One can try that. Okay, if you didn't try it, the type of data in CBio portal. Now let me take you to the OMIM. If a patient is suffering uh, from a cancer because of hereditary, okay, then obviously one should have a knowledge on what are the most important genes, okay, which is causing the cancer because of heredity. Okay, so that information can be obtained from OMIM. So you can search for a particular cancer, and or even you can search for a particular gene, and you check the more information on that. Genomics Data Common Portal, GDC. For this portal.gdc.cancer.gov. Okay, so this is the information. And for the mutation data, you can also download from portal.gdc.cancer.gov. Gene codes, the cancer proteome atlas, open targets. Now go to the genomics. So genomics is an interdisciplinary field of biology focusing on the structure, function, evolution, mapping, and editing of genomes. What is genome actually, and what is the how the human genome is constructed? The human genome is constructed from 22 haploid chromosomes. Okay, that is, we have autosomes, sex chromosomes, isn't it? If you see the haploid sets of autosomes, we have 22. So 22 autosomes plus X and Y. So totally 24 chromosomes. And the 25th chromosome is the mitochondrial chromosome. So these 25 chromosomes, okay, in a haploid number, they just mapped it in an organized way in the form of a circular fashion. If anyone asks you about the shape of the human genome, you can tell this is in the form of a circular shape, not the spherical shape. Okay, so in the circular shape, all the 25 chromosomes of a human are mapped in a circular thing. Okay, so like how we write data in the CD, similarly, the human genome looks like that. Then if you are working on a particular cancer, then obviously one can also able to map the gene. That is, they can map the particular contact locus of the chromosome. So there are more details. Even the next generation sequencing, you get this is one of the important technique used even for personalized medicine for the treatment of cancer. Now let me go to the epigenetics. So if you see the epigenetics, okay, the cancer epigenetics is the study of epigenetic modifications to the DNA of cancer cells that do not involve a change the nucleotide sequence, but it involves a significant change in the gene expression in the genetic code. That is, you have heard something about types of mutations like sense mutation, missense mutation, nonsense mutation. If you see this kind of nonsense mutations or the major mutations, which results cancers. Okay, so you find there are so many epigenetic mechanisms, okay, which are necessary to maintain normal sequences of tissue-specific gene expression are crucial for normal development. So you have to, one should understand about the epigenetics, okay, to control the cancer. If you see the epigenetics in cancer therapy, the alterations in epigenetic modifications in cancer regulate cellular responses, cell proliferation, apoptosis, invasion, and senescence. Okay, so these are the various activities we can observe during the epigenetic modifications. And of course, if you see the through DNA methylation, histone modification, chromatin remodeling, and non-coding RNA regulation, epigenetics. So these things, okay, make epigenetics play an important role in tumorigenesis. If you see the epigenetic regulation of DNA, methylation, histone acetylation and histone demethylation, you can find a pictorial diagram. Here you can find so many inhibitors. Like if you want to go for DNA methylation, you can go for these inhibitors. Okay, DNMT inhibitors, some example of drugs are mentioned. And if you want to go for these kind of epigenetic modification, that is histone methylation, then they have to take HMT inhibitors. Okay, So for each and every epigenetic modification, okay, level, prescribed inhibitors are shown here. Okay, you have to go for the particular inhibitor, and even the examples of drugs are also shown here. For histone acetylation, you can find HDAC inhibitors. The examples of drugs are shown. Okay, so one should also observe infer on the epigenetic modification. There are different DNA damaging agents and targets. Okay, how the DNA is damaged actually? The DNA is damaged because of X-rays, because of release of oxygen radicals. So these are actually an alkylating agents. So you find more spontaneous processes are happening. And you can also find UV light that is from the sun. Okay, if the oxygen radicals, if you find intensive UV light, even you find a kind of genetic modifications. And X-rays also, like you can, if you see here, they also given some examples what is going to happening on because of this DNA damaging agents. So replication errors. So we have different change of bases. Okay, like A is mismatched with the G, D is mismatched with this C. Okay, vowel base pairs. You've heard about that, isn't it? Non-enzymatic methylation of DNA by cellular S-adenosyl. So these are some of the DNA damaging agents. So how the repair process is happening on. So what are the significant genes? So all these informations are shown here. So whatever genes shown here are the important targets, okay, for the cancer. If you see if you want to study about the genome, okay, so most of the cancer patients, okay, are suggested to go for genome sequencing. I told you either they have to go for the entire whole genome sequencing, or they can even go for a particular chromosome sequencing. Okay, so if you see such a kind of a large information of the human genomes are available in the Cancer Genome Atlas program. Like how we can able to map any country or a state in the world map. Similarly, if you want to map any kind of the gene and with respect to a certain cancer, then obviously one can able to go to this website. If you see here, the TCGA generated around 2.5 petabytes of genomic, epigenomic, transcriptomic, and proteomic data. Okay, so there's a big data information available. Okay, there are plenty of databases even in TCG. And if you want to know more information on the Cancer Genome Atlas, okay, you can go to this website, cancer.gov. Okay, this is the URL we use. Okay, we can find 33 different types of cancers or tumors are more explored. Okay, and even 10 rare cancers are reported. So the most important information is like you have plenty of data. 2.5 petabytes is equivalent to 212,000 DVD information or there in the public domain database. Now those patients are taken in consideration. If you see the TCGA, if you want to access any TCGA data, you can click the hyperlink. Actually, this uh, kind of a snapshot is taken from this URL, cancer.gov. So if you see here, if you want to study about the 33 cancers, or if you want to download the protein-protein interactions of a specific cancer, if you want to go for a different characterization, that is about the next generation sequencing data information, okay, all these things are present in subsequent hyperlink. OncoDB is, of course, a large database for epigenetic regulations. Like because of DNA damage, if cancer has occurred, then if one has to understand the different epigenetic process, then obviously even he can able to know from OncoDB database. So plenty of hyperlinks are shown, okay, to understand, to download more information, cancer data. Okay, for example, if they want to work on expression analysis, which is helpful for system biology approach, so one can go to this hyperlink and they can get plenty of information on expression. Related to methylation, you can go use the link and for clinical analysis and oncoviral analysis. Okay, there are different kind of sublinks also available here. Okay, to explore more on that. And of course, there is also one more database from Indian origin for a database of epigenetic modifiers, that is DBEM. Okay, of course, from the Professor Raghava has also developed a kind of a database for this. Okay, you can find the epigenetic markers, okay, chromatin modifier. Also, most information are interlinked in this database. Yeah, I think I've completed information on. Today, I've completed an information on the basics of cancer biology, the importance of genomics, and about the importance of epigenetics. So, I completed my session on today. Okay. I shared a jam file with the meeting. I don't know what for. Okay. Yeah. So I've completed my session. If anyone have any doubts, you can ask me. Do you have any doubts? Yeah. If you don't have any doubts, on tomorrow, I'll be taking you to the concepts of network pharmacology and pathway analysis. We go for various network pharmacology databases and we see how the pathways analyzed using cytoscape. If you see the OncoDB, okay, just I can show you that alone. So the I just pasted the URL, okay. So this is the homepage of the OncoDB.org. Simply here you have to feed an input like it's asking like gene symbol. So I'm going to feed like TP53, which is the most important thing for most of the cancers. I selected that, and if I work or like to work on a specific type of a cancer, for example, liver hepatocellular carcinoma, so just select the type the gene, select the cancer, retrieve genomic profile results. You get this kind of an information. Also get the expression, okay, of a normal patient is compared with the cancer patient. You can see this much expression. The TP53 gene expression is more. Okay, the red color you can find more when compared to the blue color. The normal secretion is this, but the cancer tissues secrete more. Okay, during the cancer spread. So some information on that, how to download the data, the chromosome, what is the important chromosome important, and if they want to go for genome sequencing, one has to sequence the chromosome 17 for TP52. Okay, even the locus information is produced here. So one can able to check even from the blast, okay, genome blast, they can check whether any alteration has happened or not. So this gives an idea on that. Okay, that is oncodb.org. And similarly, if you see another database, the Indian-based origin, DBEM, a database of epigenetic modifiers. Okay, we have plenty of genes information. Okay, if you want to find an information of a particular protein, so type a protein keyword or type a uniprot and you find more information on it. So default I'm selecting this. Okay, so one particular gene, you got more the details on the cell lines for a particular gene. So whatever gene I have selected, okay, fine. What is the cell line and what kind of a tissues are targeted, and more about the detailed information of the bioassays. Okay, so all these things are mentioned here. Okay, so this is the protein I'm selecting. So you can choose any protein and you can find more information. Okay, of the origin and the other information, the origin and bioassays information. So bioassays directly connect with the NCPA bioassays. Okay, so you find more information on this. Okay, so I just completed my session on these three. Okay, yeah, any doubt you can ask me now.
Uh, Myself, Hari. I just wanted to know, since you're introducing to databases of cancer, later would you let us know how to go about the take up projects, research projects, and move on with it? Which areas can be targeted, and how? What are the steps to be followed in that?
Actually, one has to read plenty of papers on system biology approach, recent system biology and network pharmacology approach. So even
Hmm.
knowing about just bringing a drugs and using molecular talking and simulation, MD simulation is sufficient
Mm.
to publish a Scopus or SAE, Web of Science
Okay.
research papers. But based on the impact factor, one has to target extensively. One has to go deep.
Okay.
They have to dig on each and every significant gene through various approaches, and then they have to come up with any kind of remedy if possible. Either they can go
Okay.
for up to the diagnosis, or even they can go with a kind of a combination of drugs, and they can check how well the drug is going to interact with this the most important genes. So the drug
Okay.
protein protein interaction, and the gene expression analysis, the virtual screening, all these things give a latest kind of approaches to publish in high impact journals.
Okay, could you just help out with the flow chart? Like, what is the best way to go about maybe say drug protein or ligand
Uh,
ligand or ligand protein?
I do one thing. I try to
Yeah,
share, okay, the few papers what I published recently in drug design and
sure,
of course in vaccine
sure,
design.
sure.
Okay, to the director, okay, Dr. Praveen Ramanujam sir, he will be sharing it to you.
No problem. Yeah, and
Oh,
just wanted to know what is the JAM file that you have shared?
no,
So, what is the
no, I did actually.
utility?
This is the file actually shared by a research scholar. I don't know. You have to go only clarify from us.
Oh,
Okay,
okay, okay,
this is
okay,
not concerned. Yeah,
okay, okay.
yeah.
Thank you,
Any other
thank you.
doubts? Okay. Good. Thank you, sir.
Um,
Uh,
so my doubt is, can we do ML related work with that gene
Yeah,
expressions?
sure. We can able to make it out, but for related to ML and AI, I'll be teaching you in session 13 and session 14. During that time, you can understand. Like, if you want to create your own database, or if you want to create a tool from the existing database, okay, to work on the data on the database, you can still make it out using the ML and AI techniques. Obviously, the more detailed sessions on user defined creations will be explained on session 13 and session 14. But one should first understand what are the existing databases and tools available, isn't it? Later on, we can able to come to know what is not there. Then you can go for designing for that alone. Okay.
Sure. Thank you, sir.
Good. Okay. Have a great day to everyone. Good night to everyone. Bye.